Okay. Sorry for this tiny delay. So I would like to start by thanking the Project Braining for the invitation. It's a real pleasure to be here, and I hope everyone enjoys. So it seems there are almost 50 students in the room, 50 people, a lot of students. So I hope everyone enjoys what I have to say. So as Cláudia said, I'm going to tell you a bit of my work on this renal cortex-hippocampus organotypic slices and the way I've been using them as a model for epilepsy research. But before going into that, there's some kinds of several issues that need to be completely clarified. First of all, what is a brain slice? So for those that are in the neuroscience area, I'm sure everyone knows what a brain slice is, but just to make sure that you do. So a brain slice is a thin section of a tissue that can be from the whole brain or from a specific brain region, and that is obtained and is maintained in an artificial oxygenated solution or a culture media. And you know that according to the way you cut this brain or brain region, you can have coronal slices, you can have horizontal slices, like if you are cutting a bagel, or if you go from one temporal lobe to the other, you can have the sagittal slices. And there are two known types of slices, which are the acute ones and the organotypic. Acute slices have usually a 6 to 12, let's say an average of 8 hours viability, making it impossible to assess long-term changes in a system. They are considered powerful preparations for investigating the characteristics of synaptic function in the brain. And as you can see here, so I have put here an example of an hippocampal slice. So the way, let's do it. I can zoom it. So you have here, this is a filter paper with acute hippocampal slices. They are always prepared in artificial cerebrospinal fluid. They are put to equilibrate, and they are usually used in an electrophysiology setup to address issues related to synaptic plasticity, such as long-term potentiation, long-term depression. So acute slices are mostly used to this type of experiments. Then you have the organotypic slices. Usually they can be from 100 to 400 micron. There are reports of organotypic slices with thicknesses higher than this, but the most used ones, I would say, that are 350, 400 micron is the thickness, the most used thickness. And regarding these slices, they can be maintained in cultures from days to weeks. And in this way, they can overcome the short viability time of acute slices. So because of this, they can be used to assess long-term structural and morphological changes. And as you can see in these images, so in this case, I have a slice. I have slices in an insert. So these slices are put in these inserts that are then placed on a 6-well plate. And then they are incubated at 37 degrees with 5% CO2 in culture media. In culture media, in a culture incubator. So what are these particular features of these organotypic brain slices? So they all preserve the tissue architecture of the brain regions that they were originated from, and they maintain all the neuronal activities. They have an intact synaptic circuitry. These organotypic slice cultures have been typically prepared from brains of animals that are at postnatal days 3 to 9, typically 1 week old pups. So brain tissues from these young ages show a high degree of plasticity, and they are quite resistant to mechanical trauma that occurs during the slice preparation. I'm not saying that you cannot prepare slices, these organotypic slices from older animals. It's definitely much more challenging, and researchers have indeed noticed that the generation that occurs during a long-term culture is indeed increased in relation to the slices that are prepared from the 1-week-old animals. Also, when we think about organotypic brain slices, they allow a very good experimental access in the precise control of the extracellular environment, because you know exactly the amount of culture media that you have in each insert. You know exactly what you did to that insert. So they are ideal platforms for exploring the molecular pathways that underlie neural physiology and also dysfunction. So they can mimic essential features that we have in vivo pathologies. And one thing that is really quite important in these organotypic brain slices that are kept in culture is that they reproduce the neurochemical responses of the brain in vivo. So in that way, they allow treatments with pharmacological agents, and they are so considered therapeutic screening systems. So we don't need to have the whole animal. We don't need to have to be concerned about complications if it enters or if it doesn't enter the brain, because when we have a slice, we have everything there. We have all the cells, we have all the neurons, we have all the glial cells, and we have all the communications between them. So it is indeed a very physiological system. Also, a virus or any kind of particle mediating transgenic expression can also be performed in this system. So we can indeed also study the function of novel genes, and we can do it in a physiological or injured brain tissue, because if you want to mimic a pathology, we just add the compounds or whatever we need to add to mimic that pathology in an organotypic slice. For quite some time, so organotypic slices exist for quite some time. They've been studied for a long time, and in the beginning they were maintained in a roller tube system. So like I have here in the drawing, so slices were kept in a glass cover slip. So you have here the media, and then you have the slice that is kept in a glass cover slip that was glued to a piece of a filter to the glass. And these roller tubes were placed. There were systems that could carry a lot of roller tubes, and that system would be placed in the incubator. The thing is that researchers realized that this was really difficult to prepare. Slices often just left this glass cover slip, so the system was really unstable, and there was a huge variability. So someone decided to do it in a different way, which was the membrane interface culture method that was first discovered by Stoppini. And in this case, you have the cultures maintained in a porous membrane. So you have like I showed you before, you have the 6-well plate, then you have this insert that contains a porous membrane. Beneath the insert, there's the culture medium. Above the insert, there's the air that is inside the incubator, and then you have the slice there. So you have an interface between the media and the humidified atmosphere. And the media provides the nutrition to the tissues, and you have the atmospheric to provide the humidification. So this system was proven to be highly stable, and this is the one I believe, I'm not going to say 100%, but definitely the most part of the researchers, most researchers that work with organotypic slices use this membrane interface system. So what can we do with these organotypic slices? So the first thing is that over the years, these slice culture systems have been established from a variety of brain regions. So I would say that maybe the first ones were the hippocampal slices. So they were used to study for quite some time epileptogenesis, stroke, trauma brain injury, beta amyloid toxicity, and Alzheimer's disease. Then someone remembered to prepare slices that contain the cortical, the cortex, the corpus callosum, and the striatum, and use those slices to address some molecular issues regarding Parkinson's disease. Regarding cerebellum slices, they have been used quite often to study the myelinating disease, as is the case of multiple sclerosis. And also spinal cord organotypic slices are often used to study phenomena related to spinal cord injury. So I just put here some, but basically when you think of an organotypic slices, indeed it can be prepared from any part of the brain. Of course, there are some regions because of particularities of their structure that are probably more difficult to prepare, that have higher variability, but theoretically you can prepare organotypic slices from all regions of the brain. Just one tiny thing regarding the hippocampal organotypic slices. So as I said, these slices, and that is because these hippocampal organotypic slices, they often developed what we call an abnormal mossy fibers system, what is usually known as the mossy fiber sprouting. And because that happens, this makes this system really prone to spontaneous epileptiform activity. So these slices, usually I say to my students, these slices are prone to excitability. So this mossy fiber sprouting, if you just Google it, it can be visualized by this histochemical TIM staining. So basically this TIM staining, it stains these terminals, this silver staining of the terminals. And I put here an image of an organotypic slices that was treated with kainate, which is a convulsive drug. And you can see that when treated with kainate, you have this strong TIM staining that goes here in the dentate gyre. So mossy fiber are basically the axons that are projected from the granular cells of the DG to the pyramidal cells of the CA3 region. So here you have the DG, and then you have all these highly strong TIM staining. And we have a piece of this in the hippocampal organotypic slices. What about molecular assays? One of the things that is indeed very appealing in the organotypic slices is that you can basically do anything with them. There's this particular assay that people use in organotypic slices to address cell death, which is the propidimutide uptake assay. It has this huge name that I will not read. And basically PI is a polar compound that is taken up by cells that have a compromised membrane integrity. When it enters the cells, it will intercalate into the DNA and it's going to emit a red fluorescence. And that red fluorescence can be taken up, can be acquired in a fluorescent microscope and can be quantified. And I've put here an example. Imagine you have here a control slice, and in this case, it is an hippocampal organotypic slice. And you can see the areas of the hippocampus. You have here the DG, you have here the CA3 area and the CA1. And upon an insult, it doesn't really matter what it was. It was, let's imagine, an excitotoxin insult that caused cell death. And you can see that PI fluorescence increased. And we can quantify both images, and we can indeed assess the increase that this particular insult did to the organotypic slice. You can also follow this PI uptake assay. You can do an immunohistochemistry afterwards. So you can have your PI staining, then you just fix the organotypic slice, and you can add an antibody against whatever you want. You can use, for instance, the NUN, and you can have all the neuron stain, and therefore you can see if a given insult caused neuronal death. But you can also check for GFAP and for IBA, and in that case, you will know if a given insult caused any type of that in the astrocytes or in the microglia. Okay. Besides that, so just a particularity. So in immunohistochemistry, in organotypic slices, the way I do it, I know there are people that do it in different ways. So usually you have the slices in this insert. So you have the slices in the insert, and basically what we do is that we're going to cut these pieces, and we're going to put these pieces, so you can see here the slices, and you have this glass slide, and you have the insert with the slice. And then you basically put an hydrophobic pen surrounding all this, and you create two independent environments. So basically, if you want to, you can use a couple of antibodies in the first one and a different antibody in the other. So what you have to do, you have to fix the slice with the performaldehyde, you have to incubate with the primary and secondary antibodies. Usually we add a nuclear staining and we mount the preparation. Afterwards, and usually after 24 hours that usually we let the slices to dry, we just go to the confocal microscope and acquire our fluorescent images using the appropriate lasers. So immunohistochemistry in organotypic slices, you can do combination of a lot of antibodies to address neurons. In this case, I've put here the example of a confocal images where you can see GFAP in green and EBO1 in red. So you have the astrocytes. You can see here, this is the dentate gyrus of an organotypic slice that is stained. You can see here the blue beneath all the red and green. So this is the dentate gyrus. And then you have the network of astrocytes, and you can all see the microglial cells. So this is just an example to show you that the images look quite nice, and that organotypic slices can definitely be used to this type of assays. Besides immunohistochemistry, you can use them for Western blot, you can use them for ELISA, and you can use them for RT-PCR. You just have to, Western blot and ELISA, you have to collect the tissue and obtain the cell lysate. If you want to perform an RT-PCR, you collect the tissue, you extract the RNA, perform the in vitro transcription, and then you just put the samples in these highly expensive machines in order to run the reactions and analyze the results. So I just want to tell you that indeed there are other assays. I believe these are the most typical ones that I've read that were performed in organotypic slices, but I'm sure that there are many others that can be performed. I don't know about flow cytometry, but in terms of these very basic molecular assays, you can do them all. They are very, very easy to have them in organotypic slices. So after giving you this tiny piece of context, I will now show you an example of a study that we have in the lab, and that is related to one of my areas of research, which is epilepsy. And you're probably thinking, okay, epilepsy, there are so many researchers working on epilepsy. Why in the world is this so important? Well, to study epilepsy is indeed very important because we have around 65 million people worldwide that have epilepsy. And despite having already 3 generations of anti-seizure medications, the thing is 1/3 of patients are still refractory to medication. So 1/3 of 65 million makes 20 million people. So despite having all these anti-seizure medications that have multiple targets within the brain, we still have 20 million people that suffer, that have epilepsy, that suffer convulsions, and that indeed have a poor life quality. So it is important to research on epilepsy. And before continuing, I have here a video. So this is a video with the preparation of the organotypic slices as we do it in Lisbon. And so it was published in the Journal of Visualized Experiments, and I will start.
Epilepsy is a prevalent neurological disease. Rhinal cortex-hippocampus organotypic slices depict evolving epileptic-like events that resemble in vivo epilepsy and can therefore be used as an ex vivo model of epileptogenesis. This system is indeed an excellent tool for monitoring the dynamics and progression of epileptogenesis, as well as for screening potential therapeutic targets for this brain pathology. Demonstrating the procedures with me will be my students, Francisco Meda and Mafalda Carvalho. To harvest the brain from a postnatal day 6 to 7 Sprague Dolly pup, first wash the head 3 times in 5 milliliters of GBSS. Working in a biosafety cabinet, firmly insert sharp forceps into the eye sockets to hold the head in place, and use thin-tip scissors to cut the scalp along the midline from the vertebral foramen to the frontal lobes. Cut the skull in the same manner and along the cerebral transverse fissure, and use curved long forceps to move the skull pieces apart. Use a spatula to discard the olfactory bulbs and to transfer the brain dorsal side up into a 60 millimeter plate containing 5 milliliters of cold GBSS. Insert fine forceps into the cerebellum and insert the spatula along the midline to carefully open the hemisphere. Use short curved forceps to carefully remove the excess tissue that covers the hippocampus without touching the hippocampal structure. Then cut below each hippocampus. Transfer one hemisphere at a time, hippocampus side up and parallel to each other, onto a piece of filter paper. Place the filter paper onto a tissue chopper with the hemispheres perpendicular to the blade, and cut the hemispheres into 350 micron slices. Transfer the sliced tissue into a new Petri dish containing 5 milliliters of cold GBSS, and use round tip electrodes to carefully separate the slices. Keeping only the samples with a structurally intact rhinal cortex and hippocampus. The DG and CA areas should be perfectly defined, as well as the entorhinal and perirhinal cortex regions. Use a spatula and a round tip electrode to place up to 4 intact slices onto individual inserts in the appropriate number of wells of a 6-well plate containing 1.1 milliliters of culture medium per well. Use a P20 pipette to remove any excess dissection medium from around each slice. Place the plate in the cell culture incubator. Change the medium every 2 to 3 days. Use forceps to lift the insert. Aspirate the medium from the corresponding well and place the insert back in place. Use a P1000 pipette to add 1 milliliter of fresh 37 degrees Celsius medium to each well. Prepare the electrophysiology setup in a closed circuit. Verify that the flow rate of the interface type chamber is set to 2 milliliters per minute and open the Carbox valve. Check the water level in the system and place a piece of filter paper into the interface recording chamber to drain any excess medium. Place a piece of lens cleaning paper beneath the frame to supply medium to the slice, and turn on the temperature controller, amplifiers, and micromanipulators. Use a syringe to load a glass electrode with freshly prepared artificial cerebrospinal fluid. Place the glass electrode into the receiving electrode, and wait for the temperature in the interface chamber to stabilize at 37 degrees Celsius. Working in a biosafety cabinet, place the insert in a 60 millimeter plate with a drop of medium. Use a sharp blade to cut a slice from the insert and place the slice in the interface chamber with the hippocampus to the bottom right. Then place the receiving electrode into the CA3 pyramidal cell layer, initiate the continuous acquisition protocol, and record the slice for 30 minutes. To perform the propydium iodide uptake assay, work in a biosafety cabinet. Lift the insert from the well and add propydium iodide in medium to a final concentration of 2 micromolar per well. Slowly agitate the plate before placing the insert back into the well, taking care that there are no bubbles beneath the slices.
When all slices have been treated, return the plate to the cell culture incubator. For immunostaining of the slices, at the end of the propydium iodide incubation, aspirate the medium from the bottom of each well and add 1 milliliter of 4% paraformaldehyde to the top and bottom of each insert. After 1 hour at room temperature, wash the slices 2 times for 10 minutes and 1 milliliter of PBS per wash, and use a hydrophobic pen to draw 2 rectangles on microscope slides. Use a sharp blade to cut the slices from the inserts and place 1 slice into each rectangle. Add permeabilization blocking solution onto each slice for a 3-hour incubation at room temperature. At the end of the incubation, add the primary antibodies of interest to each slice for a 4°C incubation overnight. The next morning, wash the slices with PBS tween 3 times for 10 minutes per wash, and incubate the slices with the appropriate secondary antibodies for 4 hours at room temperature. Afterwards, wash the slices as demonstrated, and add 50 microliters of Hoechst solution to each slice for a 20-minute incubation at room temperature. Following Hoechst incubation, wash the slices again and add 50 microliters of mounting medium to each one. Then place a cover slip over the slices and seal with nail polish. After allowing the slides to dry for 24 hours at room temperature, visualize the immunostaining and propydium iodide uptake by confocal microscopy. Rhinal cortex-hippocampus organotypic slices depict mixed interictal and ictal-like activity at 7 days in vitro. At 14 days in vitro, spontaneous activity is characterized by ictal discharges, which evolve to an overwhelming ictal activity at 21 days in vitro, with ictal events lasting greater than 1 minute. Propydium iodide uptake and immunohistochemistry against the neuronal marker NEUN revealed some neuronal death in 7 days in vitro slices that is considerably increased at 14 days in vitro in all areas of the hippocampus. At 7 days in vitro, ramified microglia with a low CD68 expression are more abundant than EBA1 positive CD68 positive reactive microglia, whereas at 14 days in vitro, in all areas of the hippocampus, EBA1 positive CD68 positive amoeboid M1 microglia exceed microglia with a low CD68 expression. At 14 days in vitro, some EBA1 positive CD68 positive cells with a hyperramification appearance can be pinpointed, suggesting the possibility of an M2 anti-inflammatory microglia phenotype. At 7 days in vitro, the expression of CD3 is barely detectable, while in 14-day in vitro slices, hypertrophic glial fibrillary acidic protein positive CD3 positive astrocytes can be observed, suggesting a progressive activation of A1 astrocytes.
Prepare renal cortex-hippocampus slices with extreme care. Make sure to remove the excess tissue above the hippocampus, as it can compromise slices' integrity during slicing. Structural deficits will negatively impact upon slice viability. In addition to the demonstrated approaches, a plethora of assays such as Western blot analysis, real-time PCR, and ELISA can be applied to this system to address specific questions regarding epileptogenesis.
Well, hi. Okay. So, are you seeing?
Yes. Yes.
Again with your notes.
Again with my notes. So display settings, right? Swap presenter and swap presenter. Is that it? It is. Okay. So I hope you've enjoyed the Jove video. So it was during COVID, so it was really challenging, but we managed to arrange everything with them. They went to IMM and did all the filming. It was a huge day, but at least now we have all this video protocol that I think it's quite pedagogic. So just to go just to tiny details of the protocol. So first of all, we need to prepare the media. So 3 different mediums are used: the dissection media, the culture media that is optimum based, and then we have the slices in neurobasal A medium from 3 days onwards. And regarding the procedure itself, so we know that we have to harvest the brain, as you saw in the video. So we adjust the shopper, we sacrifice the animal and isolate the brain. And there comes the third note that I put here. So we'll really have to remove the excess tissue here represented in figure C. We have to remove this excess tissue that is beneath the hippocampus. That's really important. So we have to do it without touching the hippocampal structure, because if you just take all this tissue to the shopper, it's just too much, basically. The slices, it's difficult for the shopper to cut this large amount of tissue. So if we remove this piece, it's easier. Then we put the tissue in the shopper and we separate the slices. And after separating the slices, we have to look at them very careful and only select the slices that look like this. So the slices need to have a complete defined hippocampus. You have to be able to see DD, the DG, the CA3 and the CA1 area, and you also have to have this curve over here that contains the entorhinal cortex and the perianal cortex. And you do not want to have cuts in the tissue. If a slice looks like this but has tiny cuts, you know that the viability will indeed be very low. So after having this, we'll go to the culture maintenance, and that's where our protocol differs from all the other protocols published with organotypic slices. So the first difference is that in this system that we have in Lisbon, the slices have not only the hippocampus, but also the entorhinal cortex, the renal cortex, which indeed was described as being very important to the hippocampal activity, to the development of the epileptic activity in particular. So we have that first difference, and then we have the second one, is that the publications that are in the literature at the moment, they describe to keep the slices in a 25% or serum, so in an optimum medium, supplemented with 25% or serum until 2 days in vitro, sorry. And then at 2 days in vitro, they say that they fully change to neurobasal A supplemented with B27, so they completely remove the R serum at 1 or 2 days in vitro. And unfortunately, I tried to do this, to do exactly the same protocol with these slices. So I removed the whole serum and just put the slices from optimum to neurobasal. Unfortunately, that didn't work. Slices simply died. No slices at all after the weekend. So usually these cultures are prepared on Tuesday. I was all happy changing the medium to neurobasal A and go to weekend. And on Monday, the slices were all dead. So something was happening, and I ended up by contacting one of the researchers that published a paper with hippocampal organotypic slices, and he said, oh, maybe we don't do exactly that. Maybe we just, let's say, we don't change the whole media. Well, not changing the whole media makes a huge difference, because if you only change half, that means that you go from 25% or serum to 12.5%. And I wasn't doing that. So because I really didn't know what people was doing, I decided to, well, let's create something here. So our culture maintenance now, the one we've established, is that the slices are kept in 25 or serum until day 3, and beginning at day 3, we gradually decrease the horse serum. So we change to neurobasal A. First, we have neurobasal A with 15%, then with 10, and at 10 days in vitro, slices are maintained in neurobasal A, supplemented with B27, and no horse serum. Okay, and they are kept for that until 20 days in vitro in an incubator, 37 degrees with 5% CO2. And then we have to go to the setup. As you saw in the video, so we use this electrophysiological setup that is equipped with an interface chamber. So this chamber is a particular one. So we have, you saw it in the video, we have also, as we do it during the culture, we have an interface with the medium going beneath the slices and the air going on the top. And we put the slices there and we record. And now I have here something that looks more or less what you saw in the video, but I would like to show you what do we do with this. So we open this program, we use clamp fit to have this recording. This is a typical recording of a spontaneous activity in an organotypic slice. And we go to clamp fit, we open the data, and we filter. So we use a Gaussian and a Bessel filter. It is just filters that are used with science. I have to confess that I'm not an expert on these filters, but is the one that researchers also use worldwide. And we end up with a signal, with a filtered signal. And then after having this, we have to quantify the events. So for that, we also go to clamp fit, but this way we're going to ask the program to detect automatically all these events. We choose a threshold above what is considered the noise and what is indeed activity, and clamp fit automatically recognizes all these events and gives a table with each event, the time that it occurs, the amplitude that it has. It gives a table like this one that we can open in Excel. And after that, we used a program that was developed in C+ language to detect the number of bursts, the average, to detect a lot of the parameters that we use to characterize the spontaneous activity. Number of bursts, the average number of events, the duration of each burst, the peak amplitude, and the frequency. So after having all this, we can have our graphs with all these parameters in the conditions that we have throughout time, without drugs, with drugs, whatever you want. One thing regarding this program is that in order to have this, the program was based in some assumptions. And one of those assumptions is that we considered a burst whenever it lasts more than 10 seconds. And we consider that the interburst interval must be higher than 2 seconds. So this C+ program has these two assumptions. Okay, these assumptions were also based in literature, so other researchers also use, as I said, for the filters, the Gaussian and the Bessel. Also, this is what researchers usually use to define bursts and this interburst interval. And we have this. So we went on to characterize our system. So in this case, slices were prepared and maintained, as I told you before, and we've evaluated the number of bursts per slice, the average burst duration, number of events per burst, average event frequency, and we realized that the maximum activity that the slices reach is indeed obtained at 14 div. So from 14 to 21, it seems there's a number of bursts per slice, the average increases a little bit, but nothing more happens to the other parameters. We went on the characterization of these renal cortex-hippocampus organotypic slices. We did a PI uptake assay in the area of the hippocampus. And now it is time for me to tell you what is the control and what is the L. So in all these graphs, we call L are the epileptic-like slices, which means that are the slices where we deplete the horse serum, so the slices that underwent this gradual horse serum depletion. Those are the ones that depict the evolving epileptic-like events. And we compare them with slices that were always kept in horse serum, slices that were always kept in the presence of 25% horse serum. And when taking those slices to the setup, and I didn't show you, but the results are published. So we saw that they simply have physiological spontaneous activity, not the activity that you just saw in the previous slide that these epileptic-like slices have. So epileptic-like slices also have an increase in cell death that was assessed through this propydiamidite uptake assay in all areas of the hippocampus. We also had an increase in the expression in the levels of GFAP, EBA1, and in the domain of the inflammation NLRP3, and also both by PCR and ELISA, so an increase in the cytokines, in the expression of IL-1 beta, TNF-alpha and IL-6, so all pro-inflammatory cytokines, and also the same results at the protein level. TNF presented only a tendency. And of all these results, so we've concluded that, okay, these slices indeed show signs of neuroinflammation, and our attention was to this. So we saw an increase, we had an increase in these epileptic-like slices of an increased expression of the NLRP3 domain. So what is the NLRP3? The NLRP3, I don't know if some of you have heard of it, is an inflammasome. It belongs to a group of cytosolic proteins that indeed are core to inflammation. They were first described in 2002 in the peripheral immune system, and they were described as being a platform for the activation of pro-IL-1 beta and IL-18. This family is composed of various members, so they all have more than one domain, so they are multiple protein complexes. We have here some of them. The name usually is given according to the sensor domain of the inflammation, and that sensor domain indeed senses pathogen-associated molecular patterns or damage-associated molecular patterns. And when it senses these events, the protein, so the complex is formed. So it means that the sensor will recruit the adapter protein. Usually the adapter protein is the one called ASC. And through the card domain of ASC, caspase-1, procaspase-1 is going to be recruited. So the complex has the sensor domain, the adapter domain, and the effector domain, that is the inflammatory caspase-1. Among all these inflammasomes, the one that really called the attention of brain researchers, let's call it, was the NLRP3, because it's indeed the one that is highly expressed in the brain and that was seen to be activated in a plethora of brain diseases. So for NLRP3, the name is given by the sensor domain, then you have the ASC and the procaspase-1, and you have the inflammasome assembled. And the thing about NLRP3 is that NLRP3 indeed senses a lot of things, is really promiscuous, as I used to say. So usually I say that. So for NLRP3, this activation involves two signals. A first signal where you have an increase in the expression of the sensor domain and of pro-IL-1 beta and pro-IL-18. And then you have a second signal that is called the activation signal that leads to the assembly of the complex. And as you can see here, there are a lot of things that can induce activation of this inflammation, such as potassium efflux from the cell, lysosomal damage that releases cathepsin B, the increase in reactive oxygen species. So all this can activate the inflammation and indeed lead to the formation of the complex. Once formed, the complex, there's a proteolytic cleavage of procaspase-1, leaving caspase-1 active, and caspase-1, well, it does what it has to do in the cell. So for one hand, it cleaves the pro-domain of the pro-inflammatory cytokines, giving rise to the active pro-inflammatory cytokines IL-1 beta and IL-18, and it also cleaves a protein that is the mediator of the inflammatory type of cell death, that is pyroptosis. So discuss them, and I'll tell you more a bit later. So remember, cell death by pyroptosis. So why is all this important? So as an NLRP3, as I said, is core to inflammation, and it is known that neuroinflammation is considered a potential target in epilepsy. Several papers worldwide indeed proved that there is a positive feedback loop between inflammation and epileptogenesis, and there are many researchers working on trying to find within the inflammatory cascades new targets for new therapeutic targets for epilepsy. And that's what I decided to do. So I decided, okay, since we have an increase in the LNLP3 domain, let's see what's the impact of inhibiting this inflammasome. And for that, we've used in our cultures this small molecular inhibitor of the inflammasome, which is the MCC 950. So this small molecule is quite specific for the NLRP3 inflammasome. This paper really describes that it doesn't touch any other type of inflammasomes, nor NLRC4, nor NLRP1, so completely specific for NLRP3. So we put it in our organotypic slices. And after testing several protocols, several exposure protocols, we ended up with this one, in which we have our culture, so we prepare our organotypic slices, and then at day 0 and at day 3, we add 10 micromolar of MCC 950. And we wanted to know what was the impact of this compound. So the first thing was making sure that it wasn't neurotoxic. So once again, we did a PI uptake assay, comparing. So in this study, so contrary to the other study, in this second study, we have, so all slices have this gradual or serum decrease. If they do not have the drug, they are called naive slices, and the ones are called the MCC 950 slices. So we've compared these two groups of slices. So in here we have the PI fluorescence in these MCC 950 exposed slices compared to the naive slices. So we used a normalization. So we normalize it to naive slices. So naive, the PI uptake in naive slices was set as 100%, and then we've calculated the ratio for the MCC 950 exposed slices. And as you can see, in all at all time points in all regions of the hippocampus, the PI uptake is always beneath the one for naive slices, and in some cases it is even quite decreased. So we can say that, for instance, in the dentate gyrus, we can say that MCC really decreases cell death at day 3 and also at day 7. And for the CA3 area, it seems that MCC 950 has an impact at day 3 that indeed is sustained in time. So in the CA3 area, we have a much less cell death than the one we have in naive slices. We decided to check. We checked the epileptic-like events, realized that at 7 days in vitro, there were some parameters of the epileptiform activity that were already decreased, namely the average event frequency and the peak amplitude, and also a tendency in the number of events per burst. And as you can see in the representative images, you do have quite a difference from the naive slices to the MCC exposed ones. And this difference becomes even more striking when you go to the 14 days in vitro. And at this time point, you do have a decrease in all the evaluated parameters of epileptiform activity. We went on with the study, decided to look at glial cells, realized that there was a decrease in the marker for microglia. So EBA1 was quite decreased since the beginning of the culture. After having the first time MCC, after 24 hours of having MCC in the culture medium, there was a decrease that is sustained over time. The same for the CD86, which is the marker for M1 microglia. And regarding the marker for M2 microglia, the CD206, it seems that there's a tendency for having higher levels of this marker in MCC exposed slices. As for astrocytes, we also saw that MCC 950 really impaired this prevented this abrupt increase that we have in the levels of GFAP in the first days of culture. So at 3 days in vitro, there's a difference. There's a difference between naive and MCC exposed slices, and indeed GFAP levels are below the ones observed in naive slices. So GFAP levels in MCC exposed slices are indeed decreased compared with naive slices. Because of this result, because we had this CD206 increase in MCC exposed slices, we decided to perform an evaluation of the morphology of the glial cells. Indeed, so all these images were acquired under the same lighting conditions, and that's why we can say this. Indeed, we have a more striking GFAP and DIBO1 staining in naive slices compared with the ones with the staining that we have in MCC, which indeed corroborates the results from Western blot. And so there was this student, this biomedical engineer student that did the master's with me, that created a macro for Fiji, a macro that allows to calculate several parameters from the skeleton of the microglia, from the cell itself, and for the soma. So basically, the macro receives the immunohistochemistry images that we obtained in the confocal. So those images are loaded in the macro. The macro automatically dissociates the colors that we have in the image, and after doing a lot of things that Anna, which was the student that Anna programmed for the program to do, we end up by having three Excel sheets, one with the skeleton parameters, another one with the cell parameters, and another one with the soma. These graphs, I'm not. So you don't need to see them very well, because the only thing I want you to show you is that we do have differences. So the gray are, but I can at least put this a bit higher. Okay, so let's put it here. So for instance, regarding the skeleton parameters, we've evaluated naive and MCC exposed slices at 7 and 14 days in vitro, and we've obtained differences, in some cases tendencies, which this is still ongoing work, between naive and MCC exposed slices. So, like I said, this is ongoing work, but even so, we could see that arborization tree, cell solidity, and soma aspect ratio were different between naive and MCC 950 organotypic slices, and that led us to think that we could indeed have a shift. So while naive slices seem to be in this, so this is a figure that shows all the different morphologies for activated microglia. So we have the ramified resting microglia, tiny cell bodies in the huge arborization tree, and then we have the progression to an active state. The rod-like microglia that are kind of like bipolar, so they have, as you can see, processes on one end and the other, kind of like a bipolar cell. And we go further. So this one is also hyper-ramified, and until the processes are completely lost, and we end up with this amoeboid shape microglia that is usually what people speak about, the amoeboid shape reactive microglia. But nowadays, we know that there are other morphologies for reactive microglia. So in our system, it seems that the naive slices go from this hyper-ramified state to an amoeboid one, while these MCC exposed slices, they have this increased in the arborization tree, and also a decrease in the cell solidity and the soma aspect ratio. So that seems that they go to this side. So either acquiring a rod-like shape, or even going, some of them probably even go to even become again resting microglia. So this is ongoing work. We're still exploring this morphological analysis. And finally, we had to look to the NLRP3 components, and as you can see here, we have a decrease in the levels of NLRP3 in MCC exposed slices, also a decrease in ASC protein in MCC exposed slices, and again, and now it's time for me to tell you just a bit more about the pyroptosis. So, like I said, Caspase-1 basically does two things. On one hand, it cleaves the prodomain of IL-1 beta and IL-18, and on the other hand, it cleaves this protein, this gasdermin D protein, which is basically a protein that belongs to a family of proteins with a high affinity to the membrane. And what happens is that when Caspase-1 cleaves gasdermin, this N-terminal piece of gasdermin goes to the cell membrane and it creates spores. And if indeed the cell is not capable of counteracting those spores, it dies through pyroptosis. So a good way to assess the occurrence of pyroptosis is indeed to have the ratio of the cleavage protein. So we did that. So we've assessed the ratio between the N-terminal of gasdermin and the full-length gasdermin, and we've observed that while it seems that over time in culture, there's an increase in this ratio in naive slices, nothing pretty much happens when we have the MCC 950. So MCC 950 seems to prevent gasdermin cleavage. As you can see, everything stays exactly the same as in the beginning of the culture, which makes sense because MCC 950 impairs the activation of the NLRP3, and the NLRP3 is indeed responsible to give rise to the active Caspase-1 and to cleave the gasdermin. So it seems that pyroptosis is really decreased in MCC exposed slices. And that is it. So I hope that you believe me that NLRP3 inhibition throughout this small molecule MCC 950 ameliorates the epileptic-like events. It also reduced the protein levels of VIBA1 and GFAP, which strongly suggests a decrease in microglia and astrocytes activation. It affected microglia morphology. As I told you, we're really exploring this shift in microglia phenotype, which will indeed be very, very interesting. And it also seems MCC 950, it prevents gasdermin cleavage, which means that in this system we have a decrease in cell death by pyroptosis. So MCC 950 indeed has a very good impact that brain organotypic slices are powerful tools to study brain diseases, as I showed you. And regarding the system that we have in Lisbon, so I hope you trust me that renal cortex hippocampus organotypic slices can serve as a model for studying epileptogenesis. I showed you that they exhibit progressive epileptic-like activity and also signs of inflammation, mirroring what is observed in epilepsy patients. And I even show you that acting upon one inflammatory cascade really reduces the epileptic-like events observed in these slices. And that's it. It's now time to acknowledge all the people involved in this work. So first of all, my place of work, the Institute of Pharmacology and Neurosciences, that is headed by Professor Ana Sebastião, and all my students, so the NeuroID team, my actual students and also the past ones, particular Ana, Leonor, Francisco, and Elsa, that really did a lot and worked a lot for this particular seminar that I've just presented. So thank you very much. Once again, thank you for the invitation. It was a pleasure to be here, and now I can answer to any questions you might have.